All right, we'll get started. I want to thank everyone for joining us today for today's SeqSpace webinar series. Today's webinar from Dr. Nicholas Navin is about breast cancer evolution through the lens of single-cell genomics. Before we introduce our speaker, I want to review a few webinar logistics. All lines for the webinar will be in listen-only mode. You may submit your questions at any time during the webinar in the Q&A or chat panel and select all panelists. Note you may need to activate the appropriate panel using the menu option found at the bottom of your screen. That's shown in this image here. Note that closed captioning is available as a link in the chat box and that this webinar is being recorded. And with that, I'm going to pass the microphone to Fred Schumacher, who will introduce our speaker.
Great, and welcome everyone. Today we have Dr. Nicholas Navin, who is an associate professor at MD Anderson Cancer Center with a joint appointment in the Department of Bioinformatics. He's a faculty member at the University of Texas MD Anderson Cancer Center, UT Health Graduate School of Biomedical Science. He is also the director of the CPRIT Single Cell Genomics Center, as well as the co-director of the Advanced Technology Genomics Core at MD Anderson. He's the principal investigator of the Navin Laboratory, which has pioneered the development of single-cell sequencing technologies. Dr. Navin's group continues to pioneer the developing novel technologies performing single-cell DNA and RNA sequencing, in addition to innovative computational and statistical methods for analyzing the resulting large-scale datasets. These methods are currently being applied to study cancer evolution in the context of invasion, metastases, and therapy resistance. In this webinar, Dr. Navin will be presenting on breast cancer evolution through the lens of single-cell genomics. Over to you, Dr. Navin.
All right, thank you very much. Let me just share my screen here. Can everyone see my screen okay?
Looks good.
Great. So thank you very much for inviting me here to speak on the seminar series. I'm going to talk a little bit about our efforts to develop single-cell sequencing technologies and then apply them to understand various biological processes in breast cancer. And so let me just go ahead and get started. So first I'm going to give a little bit of a background in the field of single-cell genomics, particularly as it pertains to cancer and cancer research. Then I'll talk about a study that we just published on breast tumor evolution, looking at reservoirs of subclonal diversity and how copy number evolves during the primary tumor expansion. And then the last thing I'll talk about today is a computational method that our group published recently called Copycat that allows you to infer copy number information from single cell expression data. So for a long time, we've been interested in understanding how a single cell, in breast cancer, this would be an epithelial cell in the duct, is able to acquire mutations and then diverge and expand and diversify, creating different genetic and clonal lineages that ultimately result in this amalgamation of different genotypes that we refer to collectively as intratumor heterogeneity. And the big question that my lab is interested in understanding and addressing is whether this genetic diversity that is generated during tumor progression is just stochastic noise, or alternatively is important for tumor progression because it provides a substrate for selection in the context of evolution, and whether this diversity is important for tumor initiation, invasion, metastasis, and I think one of the big questions is whether it's important for therapeutic resistance. So we've been very interested in understanding genome evolution in cancer, and I'm showing you 4 models here. One of the classical models was this linear evolution model, where it was thought that driver mutations had such a strong selective advantage that after you acquire a driver mutation, there would quickly be a selective sweep and the new clones would overtake the tumor mass. A more modern view is one of branching evolution, where we now know that if you if you cut a tumor at any point in time, you're likely to find multiple populations that are coexisting and co-expanding at the same time. And this raises some interesting questions about whether they're competing with each other or alternatively might be cooperating in some way to drive tumor growth. There are other models like neutral evolution, which assumes that really after the initial genetic events, it's just neutral drift and very high mutation rates occurring. And another model that I'll talk about quite a bit today, which is we call punctuated chromosome evolution, which refers to a model in which genomic aberrations and genomic instability occurs very early on, and then that's kind of geared down or even turned off after that initial point, and then genotypes emerging from that time then stably expand to form the tumor mass. So there are really three different approaches that people are using in the genomics field to understand intratumor heterogeneity as well as clonal evolution. The first method is deep sequencing, and so this involves either whole genome or exome sequencing at very high coverage, and then clustering mutation frequencies after normalizing by things like purity and copy number to identify different clones. And so this gives you kind of a broad overview of the subclones that might be present and potential mutations that might be shared between the subpopulations. And so there's a whole series of computational methods that have been developed, like sciclone, pyclone, and others, that allow you to kind of deconvolute this data and estimate the populations based on clustering algorithms. The other approach, which really became popular with papers and work from Charlie Swan's group, is this idea of multi-region sampling. And so in this type of experiment, you would sample macroscopically from different areas of the tumor, and you would sequence the genomes here, either by whole genome sequencing or exome sequencing. And then you compare the mutations across different spatial areas to understand how the tumor might have evolved. This method works well when the genotypes are spatially segregated, like shown here as region 1 and region 2, but it doesn't work quite as well when they're intermixed in the same areas, like shown in here for region 3 and region 4. And then the other approach, and the area where we've done a lot of work on, is really in single-cell genomics. Now, single-cell genomics, you can completely resolve the admixture effects that you get of different clones harboring different combinations of mutations, but you're more limited by the number of cells that you can sample overall. And so you can oftentimes maybe undersample the tumor a little bit, especially in the early days of these technologies. We've now gotten much better at doing this, and we've mostly overcome those types of issues. And so just some studies mentioned down here, some of ours are highlighted in bold on the development of these technologies. When I was a graduate student back at Cold Spring Harbor, quite a while now ago, we developed one of the first single-cell DNA sequencing methods, and this was a method called single nucleus sequencing, or SNS. It combined isolating nuclei from frozen tumors, fact sorting them into plates in a 96-well format, and then amplifying the DNA using a method called DOP-PCR, constructing libraries for sequencing. At the time, this was still in the days of 454 sequencing and some of the early Illumina GA instruments. We then sequence on one sequencing reaction or lane with just one cell. And this would give us read depth, and we would use that to compute copy number across the genome. And then we would compare individual cells to understand which subpopulations might exist in the tumor and what their genetic relationships are. And so this was applied to 2 different breast tumors, and we reconstructed the copy number evolution here. Since that time, we've continued to develop single-cell DNA sequencing methods. And while our early methods focused mostly on looking at copy number, after that, we started to develop methods to look at whole genome sequencing of a single cell or whole exome sequencing. This is really to understand mutational diversity and mutation rates in cancer. So we developed some methods to do that. We then decided that it's quite expensive to actually run those experiments. If you're sequencing the genome of a single cell, it's the price of a human genome, and so you could never really look at that many cells. So we moved to more targeted panels so that we could look at maybe 2,000 genes, but then look at hundreds of cells or thousands of cells. And so kind of reducing that sequencing space. And then more recently, we've developed methods that use tag mutation approaches to look at copy number, and they have some really nice advantages, and I'll talk about those in the next few slides. So what are the applications of single-cell DNA sequencing methods in cancer research? One application is just to resolve clonal substructure and understand what the different subpopulations are within the tumor and which mutations they share or do not share. Another is to reconstruct clonal lineages to understand the phylogeny of tumors and how they evolved over time. And so you can use this data to order the chronology of the subpopulations and their events. Another is to look at changes in clonal frequencies under 2 different conditions. For example, between a primary tumor and a metastatic tumor, a pre-treatment sample and a post-treatment sample, or you can imagine a pre-malignancy and an invasive tumor. And then one important clinical application is really to understand the co-occurrence of mutations. With single-cell genomics, you can really understand which mutations are mutually exclusive. In this dummy example here, we're showing that P10 and CHD1 are mutually exclusive, and which mutations are co-occurring. So if you look at clone 3 here, you can see it has this set of mutations over here. So, but of course, the tumor cells are not evolving on their own. They're co-evolving with their microenvironments. And so it's also important to understand reprogramming the microenvironment throughout tumor progression. And in breast cancer, we're interested in understanding how we go from a normal duct lobular network that's very well differentiated to these premalignant diseases like DCIS, where the tumor cells begin to fill the ducts, but the basement membrane is still intact. And then as you progress to invasive disease, where the tumor cells really start to spread out and invade the surrounding tissues and then eventually metastasize. And so single cell RNA sequencing approaches are really great for resolving the tumor microenvironment. When you perform an experiment like this, it's unbiased, and typically you might detect tumor cells as well as immune cells. T cells, B cells, and myeloid cells are quite common. And then also stromal cells. So this could include endothelial cells like vasculature or lymphatics, fibroblasts, and other cell types. But in addition to just resolving the cell types, the powerful thing about single cell RNA sequencing is that you can then take a group of cells and you can recluster them to get very high granularity and identify different cell states or biological expression programs that imply different functions. With T cells, these states are pretty well described, but there are always new ones being detected now with these methods, like CD4s, Tregs, CD8 cytotoxic populations, et cetera. But then when you go to, for example, fibroblasts, it's much lesser known what the different types of fibroblasts are. So these methods are very good for classifying cells. You can also look at the tumor cells, so you can get their expression programs and then really understand how different phenotypes might vary within the tumor population. For example, which cells are more mesenchymal or epithelial, which ones are more hypoxic, which ones are proliferating more, et cetera. You can also do a subtype analysis to see how many different of the cancer subtypes or signatures are kind of coexisting within one patient. You can reconstruct lineages to understand development in tumors where these developmental trajectories are still intact. And you can do cell type specific differential expression analysis, which is really powerful because you can then kind of zero in and just ask, how the fibroblasts affected in response to, say, chemotherapy by looking at pre or post-treatment samples. So if we take a look at the papers that have been published in the field of single-cell genomics, a lot of the initial papers focus more on looking at the genomics of the tumor cells and how they evolved, as well as the microenvironment. Following that work, more studies started to focus on metastasis and circulating tumor cells, work by Dan Haber's group and others, some of our papers as well. And then more recently, I think the two areas that are just kind of really growing, and I think they're very exciting, one is to look at pre-malignancies to understand how these early diseases progress to invasive diseases. Lots of challenges here, of course, with the limited number of cells that you're dealing with and FFPE materials and so forth. And therapeutic resistance, where you're trying to look longitudinally at how therapies are impacting cancer cell populations and the microenvironment. And here also many challenges with very limited samples in terms of core biopsies that you have to work with to do the single cell profiling. But we're generally very excited about some of the translational applications of these methods. We think they can impact many different areas in terms of clinical diagnostics, as well as improving therapeutic strategies. We think these methods could be very good at early detection. So you can imagine for bladder cancer in the urine, maybe detecting early cells that have mutations or copy number changes. You can imagine in the sputum or in your spit, maybe detecting early lung cancer cells. Many opportunities there. For risk stratification, so trying to figure out which patients are more likely to progress and which will more likely to stay in situ. Matching patients in clinical trials, predicting therapeutic response. And then really, we think these tools are quite useful for discovering a lot of new drug targets, not just on the tumor cells, but on the microenvironment, because there really are unbiased methods. All right, so that's a little bit of kind of introduction and background, and I'm now going to move on to talk about the two research studies that I mentioned earlier. So the first study is on breast tumors, that triple-negative breast cancer, and how they maintain copy number events and evolve over time during the expansion of the primary tumor mass. And this was a study that involved a graduate student in my lab, Darlene Manussi, another graduate student, Hongwei Yi, and a collaboration with Francisca McCord, who is at Dana-Farber Harvard, one of our postdocs, Michael Nicholson, and then also Peter Van Loo, who's over at the Crick Institute in the UK. So we've been interested in some time now as to how a normal diploid cell, shown here in this copy number plot, can acquire chromosomal changes and have complex aneuploid characteristics such as chromosome amplifications and deletions across the genome, and then how it can kind of maintain this genomic profile as the tumor mass expands to millions of cells. And so a while ago, we started investigating this and trying to understand, at least at the single cell level, whether a gradual model of copy number evolution was more likely, in which you acquire one amplification, then a second one, and then maybe a third deletion and a fourth one, gradually and sequentially over time, as shown over here in this plot, or an alternative model, which we call punctuated evolution, borrowing this a little bit from species evolution, where it's been published by Stephen Jay Gould and others that there's a punctuated equilibrium model, that there's this initial burst of instability, and then you acquire lots of chromosome aberrations, and after that, the genome is relatively stable as the tumor mass expands, and that's shown over here in this plot. Now, the paradigm for copy number evolution and also for mutations has really been a more stepwise series of mutations that occur over time. And this was really popularized in colorectal cancer by Bert Vogelstein and Eric Ferron, showing that at least in colon cancer, there was this stepwise order of mutations and copy number events that occurred, leading from the very early stages of the adenoma, of normal epithelium through adenoma and then through adenocarcinoma, with mutations in APC, then KRAS, then P53 being a late mutation. A lot of these ideas extended to breast cancer, and especially a lot of the cytogenetics work early on, mostly from DNA fish experiments, but then later through microarrays and other experiments, suggested that there was really an ordered series of chromosomal events occurring throughout breast cancer. But as we started to look at single cells in breast cancer, we found that there just are not a lot of gradual intermediates. And if you try to model it mathematically, it seems very improbable that those clones would emerge such that there would be a selective sweep during tumor progression that would lead to a very stable copy number profiles. And so work from our lab has published over the years on what we call punctuated chromosome evolution, where we see support for this burst model, especially in the triple negative breast cancers. But one question we had about our data is that a lot of the early single cell methods were good at sampling a small number of cells, but really couldn't look at many, many cells to kind of understand what's happening over a large time frame. And so the big question for us was, although our data supported this early punctuated model of copy number evolution, what happened after the initial punctuated event? Was the genome completely stable and just expanded as this identical genotype through symmetric divisions, or maybe even asymmetric divisions? Or was there still a continued copy number evolution, just at a much lower rate? And so that's what we set out to investigate in the study. To do this, we had to develop some better single-cell DNA sequencing methods, and we also wanted to have single molecule resolution so that we could really measure accurate copy number events. So the method that we developed is called acoustic cell tag mutation. We dissociate a frozen tumor into single nuclei. We flow sort them into plates. This can be done in 384 well plates. We've also tested it in 1536 plates. From there, we use this system called an acoustic liquid transfer system. And this allows us to do two things. One, it allows us to scale down the reactions to really nanoliter volumes, such that the total volume is less than a microliter when you're finished doing experiments. Second of all, it automates everything. So it has robotics that allow you to really go through and do this in a very high throughput way. And so this really reduces cost quite a bit. The sound waves allow you to transfer liquid of about 10 nanoliters or so from one source plate to a destination plate. And so the destination plate here has the single cells sorted into there. And we do a reaction here called a tag mutation reaction, where we're taking the DNA, we're doing a DNA tag mutation, which means we're fragmenting the genome, and then at the same step, we're adding adapters on there. And then we do a PCR amplification using dual barcodes, and we have these dual barcoded libraries that we just pull in the end to run our sequencing reactions. And so compared to our previous DOP PCR-based methods, which took about 3 days to complete, these whole experiments can be done in about 3 hours. And the chemistry is very simple. It's 3 steps, uses commercially available TN5 transposases, and has pretty low costs associated with it. Now, of course, we were not the first to think about doing tag mutation-based chemistries in single cells. There's actually already some existing literature on this. In fact, one of the papers by a colleague of mine, Andrew Addy, was published a long time ago, showing that if you dilute DNA down to concentrations that are similar to what you would see in a single cell, let's say 10 picograms, you can construct very low input sequencing libraries from that material. And then subsequent work at Fluidigm by Richard Facchetti, and then later on by Saurabh Shah, Sam Aparicio, showed that you can use tag mutation-based approaches to make single cell libraries and then kind of pool them and sequence them to look at copy number information. And so that work was also, that approach has also been used in a method called combinatorial indexing, also by Andrew Addy's lab, where they do multiple rounds of flow sorting and barcoding to try and barcode cells and do tag mutation. There's also an approach developed by 10x Genomics. It's actually not tag mutation based. It's based on a whole genome amplification chemistry. But unfortunately, they've discontinued the system as of a few months ago, or this year actually. And so I think right now, really the leading approaches to do single cell copy number measurements is going to be by tag mutation. So this is just to show you quickly one of the advantages of tag mutation, which is that if you do DOP PCR, you're basically priming off of existing strands that you've already amplified. And because of this feature, you can never determine where the original cut sites were in the genome. That's different than what you do with the tag mutation approaches like acoustic cell tag mutation, where you do that initial tag mutation, you're introducing the barcodes there, and then you're amplifying just those fragments. And so you preserve those initial cut sites. And so by using something called positional barcoding, you can then get rid of all that over amplification. And ultimately, you can go back down to those original diploid molecules so that you have a DNA count of only 2 for every site of the human genome. Or in cancer, you might have 3 or 4, depending on the copy number. But you eliminate lots of amplification bias, and you essentially get what's equivalent to single molecule data. So we tested out our method and kind of benchmarked it against other methods. And we saw that our ACT method showed an improved performance in terms of the total coverage breadth that we get across the single cells compared to the 10x platform, also to the DLP platform from Saurabh Shah, and especially compared to our older DLP PCR-based methods. And we also looked at overdispersion, which is kind of the measurement of noise going from the segment of the chromosome copy number to kind of the bins that would be dispersed around it. And we found that really it also showed improvement in the overdispersion compared to the other methods. This is just an example profile down here of a single cell. You can see the binned data across the genome and the segments, and you can see the copy number states are quite clear here from this data of a triple negative breast cancer. So we applied this method to sequence about 1,000 cells from 8 different triple negative breast tumors. They were all untreated except for one case, TN1, which it was treated with AC. And many of the tumors, when we sorted them, had higher DNA ploidy, so in the range of triploid to even tetraploid, suggesting that likely there was a genome doubling event that occurred in most of these tumors. So I have to explain a few definitions here in talking about this data that we often use in our lab. So we refer to subclones as individual populations of cells that share what we would call an identical genome or copy number profile. And we refer to superclones as a grouping of those subclones that share a number of copy number events, but are also distinct from other superclones. And so if you look at this high dimensional map right here, the UMAP space, these are the superclones, and then you can see the subclones are kind of these little groups within there. Each dot here represents a single cell. And so we have different classes of copy number alterations as well, and we call these clonal copy number events. Those are common across all the subclones, or at least 90%. Subclonal events, that's in less than 90%, but more than 2 subclones. And then unique copy number events, which occur only in one individual subclone. So just to explain what that type of data would look like, here's a clonal event, a copy number event. So these are all subclones going down here. These are 2 genes, FGFR4 and P53. And you can see they all have the same copy number. It's 2 copies for P53, 4 copies for FGFR4. If we look at subclonal copy number events, you can see that about half of the cells roughly here have this amplification of cyclin D, and half of them have this PIK3CA amplification. If you look at unique events, and this is really what we now have the power to detect with the current methods that we're using, you can find that things like, if you look at EGFR, there's only a single subclone that has this deleted, or amplified, sorry. If you look at MYC, there's only a single subclone here that has a lower copy number. So we applied this method to look at 8 triple-negative breast cancer patients. And this is showing you the superclones and subclones that we detected in each patient. You can see there's quite a range. And here we show the superclones kind of ranging from about 3 to, I think, 5 or 6. And if you look at subclones, you can see there's quite a range too, going down from 6 all the way up to 22 different populations. And if you look at the copy number counts, we can see that there's also a range here where pretty much every tumor, of course, has clonal events, subclonal events, and unique events. But again, we see kind of a range, and some have fewer and some have lesser events. So let's look at the data here for 2 tumors. This first one is going to be the first triple-negative breast cancer. You're looking at about 1,100 cells here going down the y-axis. And you see the color bars over here represent the superclones, and over here, those are the subclones. And so you can see, for example, within a superclone population, you can see a lot of larger genomic events that are shared. If you look at an individual subclone, you can see that there are also specific events for each subclone that kind of define that population. And so this individual tumor here, we see highlighted down here some of the different genes that were classified as being clonal, subclonal, or unique. And so, for example, I think I showed these before. There's FGFR4, P53 are clonal, where cyclin D and PIK3CA are subclonal. And here there's the EGFR amplification that is unique, and a MYC loss that's actually unique to one population too. Here's another tumor, TN2, again, looking at around 1,000 cells. You can see all the different subpopulations that are present here. And if we look at the genes down here, you can see that just a few select ones that we pulled out. If you look at the clonal events, things like progesterone receptor and PTEN were kind of clonal throughout the tumor, whereas EGFR was subclonal, and about half the populations had this amplification. And and PIK3CA, where there are just a few cells that had a lower copy number compared to the rest that all had copy number 6. And then here are some unique events where just a single subclone has the amplification. So next we wanted to take this data and reconstruct how copy number evolved over time. And to do this, we also wanted to perform exome sequencing to have the mutational data as well in order to really estimate genome doubling and look for other mutations that were happening during evolution. So we performed exome sequencing of the 8 tumors. We found that there was really only one mutation that occurred at a high frequency, and that was P53 mutations, which are, of course, known to be associated with about 83% of triple-negative breast cancers. We then took the exome mutations and we classified them based on their frequencies as being either clonal or subclonal, as you can see over here. We then took the clonal mutations and we used those to estimate the whole genome doubling events, looking at LOH and when those events occurred, and estimated this for all the tumors. We found that the whole genome doubling occurred roughly about half the time between that initial diploid cell and the most recent common ancestor shown over here. We also timed the P53 mutations relative to that whole genome doubling event and found that in all the cases, P53 occurred prior to whole genome doubling, suggesting that it might be important for the whole genome doubling event to occur. And then we call this a truncal distance. This is kind of the initial distance that occurred prior to the real divergence of all the subclones I showed you in the previous slides. So in our previous data, we always were able to identify this long truncal distance. But as you can see here, there's also a distance occurring after the MRCA to the last nodes. And we refer to this as the branching distance. This is really what we can now resolve better with our methods. Here's another tumor here. You can see there's a truncal distance, but there's also a fairly large branching distance occurring after that most recent common ancestor. We're now kind of zooming in and looking only at the copy number evolution occurring after the MRCA. And instead of showing you single cell data here, we're showing you a consensus of the clonal genotype of the groups of single cells. And in this particular case here, we see that there's this splitting of 2 major lineages, A1 and A2, and then they continue to evolve and diversify into the major superclones shown here in the big colors, and the subclones shown over here in kind of this rainbow of different colors on the side here. And same in this patient here, we see initial split of these 2 lineages and continued copy number evolution occurring as the superclones diverge and then the subclones within those superclone groups. We could actually use this data then to compute what the most recent common ancestor looked like by looking at the minimal profile between all these events, and that's kind of shown over here and over here. And so this data really shows that there is a lot of copy number evolution ongoing after the most recent common ancestor, as the tumor mass really expanded from just the initial few cells to millions of cells that constitute the final tumor mass. Next, we collaborated with Francisca McCord and one of her postdocs to ask another question about this, what we think is a punctuated chromosome evolution model. What we wanted to know was, after the initial punctuated burst, did the copy number evolution rate just decrease dramatically, or was there another period with increased transient instability that occurred prior to this leveling off of the copy number evolution rates? And so they developed this binary tree branching process, and were able to model the growth of the tumor over time. And then after this, using these two models, so one where the copy number rate was flat and another one where it was elevated and then dropped down, this is after the initial punctuated burst, they were then able to run these simulations and look at the chromosome breakpoints that occurred from this. And then they were able to compare the chromosome breakpoint frequency spectrums of the model data versus the experimental data that we had for each tumor. And what we found was that in pretty much all the cases, we saw that the chromosome breakpoint frequency spectrum was matched much better for us for the transient instability model. Excuse me. This is the model where there's an increased rate of copy number evolution directly after that initial punctuated burst, and then it drops down. And we saw this was matched very well for most of the tumors, as shown over here by looking at the IC scores of the GA model minus the other model, the transient instability model, for most of the tumors. Now, there was one tumor, TN8, that was technically fell on the kind of right-hand side of the graph. This one we're not 100% sure of, but most of them look like they're pretty consistent with this idea of transient instability. So the next question we asked was, what do triple-negative breast cancer cell lines look like? Are they very clonal because they've been passaged for a long time in culture, or do they retain the genomic heterogeneity that we see in these triple-negative breast tumors? And so we applied this to 4 different triple-negative breast cancer cell lines that are very commonly used. And we found, indeed, that in these cell lines, we could also detect a very large number of subclones and superclones that was comparable to what we see in the human tumors. And of course, also occurred in many genes that are relevant to breast cancer progression. We next performed an experiment to try and really get at the question of what happens to the single cells as you subclone them and then expand them for a known number of cell divisions. So we did this for 20 cell divisions to get back a plate of cells after subcloning individual cells. And then we performed the single cell copy number profiling again. And what we found very striking here was that we didn't see a completely clonal genotype after we did this. Instead, we saw that the subpopulations emerged again after only 20 cell divisions and kind of re-diversified the tumor mass. And so that was quite interesting. We think it has important implications for people who use isogenic cell lines, at least in studying triple-negative breast cancer. And we could also estimate what the copy number evolution rate was, which was about 1 copy number event per 4 cell divisions here. Next, we want to understand, does subclonal copy number impact gene expression programs, and then ultimately impact the phenotype of the cell? And so we took these single cells, we isolated them and expanded them, and then we got kind of a bulk expansion of those cells, and we performed bulk DNA sequencing and RNA sequencing. Now, I told you before that doing this actually creates more genetic diversity again after the expansion. But when you look at the average of everything, it still represents the initial population from which you were able to subclone the cells. And so doing this, we were able to look at the expanded subclones. And if we look at the copy number profiles shown over here, overlaid with the single cell data, copy number data, we got most of the subclones represented very well. I think it was about 10 out of 11 major subpopulations. They're shown over here. These are the bulk expanded clones, and you can see how they represent the different subclones and superclones. If you just take the RNA-seq of the expanded subclones, they can also separate pretty well, at least into 3 major populations, reflecting the superclones. We next asked globally, how does the gene expression correlate with the copy number? And we saw that there was pretty much a linear relationship here, where as you increase in copy number, you increase in the overall gene expression. We next looked at various subclonal events and tried to understand, do they impact gene expression, especially of breast cancer genes in those regions? And so this is showing you a copy number plot here now for each subclone. And these are two subclones that had this amplification here on chromosome 11q. And that increased the copy number for ATM and CBL. And if you look over here at the gene expression, you can see that those two subclones also increase in gene expression for these genes. If you look at a moving average windows, this is not looking at specific genes, but kind of looking at all the genes across this interval, you can see that those two subclones, the gene expression increased quite dramatically here in this region where the subclonal copy number occurred. Here's another example of that. This subclonal copy number event occurring over here. And so this is for PIK3 R2. And you can see over here in the gene expression plot that that area is also increasing gene expression. You can also see it over here with the moving window. So next we looked at signatures because we wanted to understand beyond the just subclonal copy number events, how is the whole transcriptome being affected both by cis and trans events that might occur as a result of these subclonal events. And we saw that it really did impact quite a large number of signatures that we're interested in. These are the top ones that we identified. You can see how they differ across the different subclones that we expanded in culture, suggesting that the subclonal copy number events really might impact the larger cancer phenotypes, things like EMT and hypoxia, and diversify those genotypes and the phenotypes within the population of tumor cells. So to summarize this part of my talk, what we see here is that we don't really see evidence for a gradual evolution model, at least not for copy number events. We do see, we do see support for what we saw previously, which is punctuated evolution. And so we think a lot of the copy number events are occurring early on in this initial burst of instability. But previously we thought that later on there was this period of clonal stasis where there wasn't much copy number evolution occurring during the expansion of the tumor mass. And this model was mostly, I think, due to the early single-cell sequencing technologies and also the limited number of cells we could really sample here. Using the newer techniques that I showed you today in the talk, we now infer this model where there is punctuated chromosome evolution, but importantly, there's this period of transient instability occurring after that, where there's quite a lot of genomic diversification and ongoing copy number evolution. And then following that, although some major clones are then selected and expand, there's still a level of ongoing copy number evolution that's occurring at a lower or reduced rate. And so this is our new model now for how copy number evolves in these triple-negative breast cancers. So just to summarize this part, we developed a single-cell DNA sequencing approach. It's high throughput and has single molecule resolution. We were able to detect clonal substructure in these triple-negative breast cancers, and we found that there was a very large number of subclones organized into just a few major superclones. In our previous studies, I think we had the power to detect superclone populations, but not really the subclones. We see that in evolution, you have this early p53 mutation. Following that, there's this genome doubling event. And after that, you get this period of punctuated chromosome instability where you get a lot of genomic diversification. After that, what we see is that then there's ongoing copy number evolution and this period of transient instability that, again, continues to diversify the copy number of the tumor as the tumor expands. And then finally, we think this is an important finding, and we've seen this in a few cell lines now. It really doesn't look like procedures such as isogenic expansion can produce clones that have the exact same genotype. Even in a relatively short period here, about 20 cell cycle divisions, we're getting a big rediversification of the genome or the copy number profile of those individual cells, at least for these triple-negative breast cancer cell lines. I think we'll have to see how this pans out in other cancer types and other cell lines, but at least here we think it's really not possible to produce an isogenic population using these kind of standard procedures. Okay, and the last thing I'm going to talk about today is a method that was developed in my lab by a postdoc, Ruli Gao. She's now an assistant professor at Methodist Hospital, and this method is called Copycat. It stands for copy number karyotype analysis of tumors, and this was published a little bit earlier this year in Nature Biotech. And this was a collaboration also with two other colleagues at MD Anderson, Stephen Lay and Stacy Mulder. So the problem is that we often do single-cell RNA sequencing experiments, and it can be difficult to determine where the tumor populations are. You can look for things like EpCAM expression, but oftentimes you might find two clusters of cells that have high levels of epithelial markers, and you don't really know which of these is the tumor cells. And so we wanted to take advantage of aneuploidy because many tumors exhibit aneuploidy. And we wanted to be able to use that to say this is likely to be the tumor population, and these other ones are likely to be stromal or immune populations. because they would be diploid. Now, there has been some previous work on this, and there was a really nice work done by Itay Tirash, working also with Alex Schlecht and others. And they showed that in single-cell RNA data, if you use a moving average window, you can detect aneuploid rearrangements, and you can kind of classify the tumor cells as being diploid or aneuploid. But this method also has some limitations. So really, you have to provide a matched normal reference each time, and so that is required, and it's kind of a step that makes it more difficult to run these analyses. The moving average window, at least in the initial versions, is kind of a very simple way of just looking at number of genes and how they're expressed over the genome. But it's hard to get real definitive chromosome breakpoints, which is something that we're very interested in. And then the hierarchical clustering they use, it doesn't really resolve the clonal substructure that well. And so these are reasons why we wanted to kind of continue working on and developing these methods. So this is a Copycat, and in the interest of time, I'm going to go through this a little bit quickly, but basically we get our cells, and we have our genes and an expression matrix. We remove some outliers in the data. We then do this kind of hierarchical clustering, and we use this Gaussian mixture model to identify which of the clusters is likely to be diploid cells within your sample. And then based on that, we run a segmentation approach, which uses an MCMC, and it uses Kolmogorov-Smirnov to determine whether two segments should be joined or not. And this gives you segments, which is equivalent to copy number events with accurate breakpoints. We then try to identify what is likely to be, based on our transformed gene expression here, which cells are likely to be diploid cells and which cells are likely to be aneuploid cells. We then take the aneuploid cells and we perform additional clustering where we can identify then subpopulations. And shown in this tumor here, there's two major subpopulations labeled A and B. We then take those subclones within the same tumor, and then we kind of run things like differential expression analysis, signature analysis to understand their differences. And we can go down to about 5 megabase resolution with these methods, which we think is pretty good, and it's compatible with high throughput 10x based single cell RNA data. So we applied this method to about 20 tumor samples, including both 3' and 5' data, including 5 anaplastic thyroid cancers, 5 triple negative breast cancers, and 5 PDACs, and also 2 ER positive breast cancers. And we also looked at some of the older single cell RNA sequencing methods based on the SMART-Seq2 chemistry. And so this is just showing you some data here comparing the DCIS in a DCIS tumor, the Copycat inferences of that data. So you can see over here, this is the copy number profile, to what we consider a gold standard here, which is a bulk DNA sequencing profile over here. And this is infer CNV, so this is the other method. You can see it's kind of, because it's a moving window, you get kind of these wavy curves across the genome. The pattern does look, quite similar. It does reflect kind of read-based density. But we saw an improved performance for our method, Copycat, both in kind of the relative deviation to what we consider to be the bulk standard sample, as well as the kind of stability of the intervals across the genome. So we applied this to 5 pancreatic ductal carcinoma cancer patients. This was from a published study by Peng et al. 2019. And in the study, we could identify the expression. This is the RNA data up here. And then these are the EpCAM markers down here. And this is showing you data from one patient here. These are the tumor cells that are identified, plus their copy number aberrations. And these are the diploid cells over here. And so when you infer aneuploidy, you can see in the expression data, you have 2 clusters that have epithelial markers. But with aneuploidy, you only get one of those clusters, as shown over here. Again, in this patient, there's 2 epithelial clusters. Here we can identify the one cluster that's the tumor cells. And that was the case for the other tumors as well. These are the 5 anaplastic thyroid cancer samples. You can see that the tumor purity is actually quite a lot higher here based on our aneuploidy detection. And these are just examples where you can see the expression of oftentimes 2 clusters, where one of those we detected as aneuploidy. And so we think this method works quite well, at least in these tumor types. We also applied this to triple negative breast cancers. And this just shows you some more examples. Oftentimes with the triple negatives, we could identify multiple subpopulations that had aneuploid rearrangements, suggesting that both subpopulations were likely tumor cells, and there were multiple subclones in the tumors. Also, we could estimate the tumor purities, which you can see are much higher here for the triple negatives compared to, for example, the PDACs that I showed you earlier on. And so we got a very high accuracy based on classifying the tumor cells based purely on the copy number and the aneuploid profiles for these cancer types. I can tell you right off the bat that this approach does not work that well in pediatric cancers, also in liquid cancers where there aren't many copy number changes. But at least for these tumor types, we got pretty high prediction accuracies. We then went further and looked at subclones within the tumor, and this is just showing you a triple-negative breast cancer patient where Copycat detected 2 different subpopulations. We compared these are kind of the consensus copy number profiles, and you can see they share a lot of events, but really interestingly to us, there's a major KRAS amplification here in chromosome 12p12.1 that was amplified in one subclone and not the other one. And we did a differential expression analysis between the 2 subclones, identify a number of pathways that were different between them, and importantly showed that KRAS was really high in this kind of rare subclone within the tumor, whereas the other clone didn't have that KRAS amplification. And KRAS amplifications are actually not widely reported in triple-negative breast cancer, so we thought this was quite interesting. Here's another tumor where we also detected a KRAS amplification. There are 2 subclones inferred from the single-cell RNA data labeled as A and B. Here's that KRAS amplification you can see over here. See in clone B, the levels are higher as well. This is significant. And we did differential expression analysis, and this kind of links the genotype to the phenotype and allows us to look at some of the pathways and signatures that were different between these subclones. So just to summarize this, we think this is a kind of a neat tool to be able to infer copy number from high throughput single cell RNA data. We have about a 5 megabase resolution. And at least in the cancers that we looked at with that had solid tumors here, we got a very high prediction accuracy based on the aneuploidy alone, not using the gene expression information to classify tumor cells and normal cells. And this software is all open access, and anybody is welcome to use this. And the link is down here if you're interested. So just in the last minute or two, I just want to mention some exciting, I think, future directions in the field of single-cell genomics. One of them, I think, is really to be able to use single-cell methods. Mostly I'm talking about RNA or attack-based methods or even methylation-based methods to define organ systems and have a reference of what normal is, so that when you start to study diseases like, in our case, DCIS or invasive breast cancers, you know what normal looks like and how that varies across individuals. So we're very active in projects like the Human Cell Atlas. I didn't talk about it today, but we're working on the Breast Atlas, and also very excited about projects like HubMap that are really looking at what normal tissue looks like prior to going into the disease setting and defining how different cell types are programmed. We think single-cell multiomics is also a very exciting area for active development. In our case, we're really interested in how we can link DNA information to information that we would use to quantify the phenotype, which would be RNA or epigenomics. So we think these methods can really link the interactions of mutations, copy number events, to the final output, which is the expression program or the phenotype of the cell. Some progress has definitely been made in this area. There's some methods like GNT or GOT or a few others. They're still fairly low throughput and still have a lot of technical errors. So I think this is an area that we still need to really be able to work on to make high throughput and robust. And then another area is really marrying the single cell genomic methods with spatial resolution. And we need this because, of course, in cancer, you want to look at histopathology and understand how different morphological features are linked to the genotypes or the phenotypes of the cells. And so, of course, there's a lot of development in this area right now, but there's still not the perfect solution, I think. There's spatial genomic methods that are unbiased, like spatial transcriptomics, 10x genomics, or Visium or SlideSeq, but they don't have single cell resolution. You have to lyse the tissue and then hybridize the probes. Currently, they're around 10 to 50 cells or so. And then there's in situ sequencing technologies, which allow you to get single cell resolution, but then they either don't have kind of large spatial resolution, or a lot of these are limited to the number of transcripts that you can actually look at for reasonable, economical cost in an area of the tissue. So we think this is a very exciting area and still one that needs to go pretty active technology development to get to kind of the perfect, I think, balance of having unbiased genomic profiling with single cell resolution in tissues. And then finally, an area that we're really interested in is being able to make single cell DNA sequencing methods or RNA sequencing methods that are compatible with archival material. At least here at MD Anderson, we have lots and lots of samples with long-term clinical outcome. And right now, they're just not compatible with single cell sequencing methods. So we're doing quite a lot of work in this area to try and develop methods to work with FFPE materials and archival materials to be able to do single-cell DNA profiling, and work with these kind of fixed materials which are routinely collected in pathology. So that brings me to the end of my talk. I just want to thank all the people in my laboratory who did the work that was presented today. Darla Manusi, Hanghui Yi, Shen Shen, Emmy Say, Ruli Gao, Alex Davis, Tapsi Seth, all of our wonderful collaborators here at MD Anderson, Harvard, Crick, and other places, and all of our funding support, especially from the NIH, our one grants, and everyone else is kind of supporting this work. And I think I can stop it here and take any questions that you might have. So thank you.
Thank you, Dr. Navin. This is a really nice overview of the approaches to single-cell sequencing and their applications that can inform our understanding of cancer, particularly for copy number alterations. I just want to invite participants to submit their questions in the Q&A box. And while we wait for those, we have some already. It sort of relates to your second to last slide, and there's just a question about if you could comment on the tissue types used or that are preferable to use for single-cell sequencing. And in the case of your triple-negative breast cancer study, did you focus on a particular tumor stage?
Yeah, so I guess the first question is more general. What types of tissues can you use to run single-cell sequencing? It really depends whether you're talking about single-cell DNA sequencing, RNA sequencing, or epigenomic profiling. For single-cell RNA sequencing, you really need fresh tissues to work with, viable cells. That's going to give you the highest quality data. You can run single nucleus RNA sequencing on SNAP frozen tissue because the nucleus kind of protects the RNA there, but the plasma membrane gets lysed. And so those experiments are possible, but in my opinion, the cell data is much better when you can obtain it. Single-cell DNA sequencing works fine with frozen tissues or fresh tissues. DNA is quite stable, and if you isolate nuclei, you can really work with that material. Now in FFPE tissues, that's where it becomes more challenging, and so we're working a little bit on methods to try and repair DNA and reverse cross-links and things like that to be able to do the profiling and get high-quality data back. Yeah, so I think that's just about the tissues. And the other part was about the triple-negative breast cancers. What stage or grade were those? So these were mostly treatment-naive patients, and so they were all high grade, but they were not metastatic at the time that we collected them to analyze them. And yeah, so that's kind of where they are. They were all untreated because we wanted to look, a bit more here at the kind of natural genomic diversity of those tumors. There was just one patient that was treated with anthracyclines. That was TN1.
Thank you. You mentioned you're doing work on FFPE samples, and as you said, there's a lot of archival specimens in that area. So do you have an idea of sort of how far things are along in terms of method development and when we might see some approaches where we could do single cell for FFPE tissue?
Yeah, absolutely. I think for DNA, us and other groups are working on this now, and so those approaches will be coming online pretty soon. We have some papers that we're working on right now, and technology development. There is highly variable, depending on how old the block is, and if you're talking about FFP blocks that might be 10 or 20 years old, then the DNA is highly degraded, and there it's really important to employ all types of methods to try and fix the DNA before you do these assays. If you're talking about more recent blocks that have been embedded, especially ones here that are fairly high quality, really not too much DNA repair is necessary, and you just need to deparaffinize those tissues. So yeah, I think those are methods that we're working on. We'll be able to report back to you on pretty soon, and I think other groups too. A lot of people are really interested in using them because of the long-term clinical outcome that you can tie to those types of samples.
Right. Yeah. The number of cases included in single cell sequencing studies right now is often small or smaller than like the bulk DNA, RNA sequencing. Do you think there's a point where we start to saturate the information gained from single cell sequencing data in terms of sample size, or do we just need to keep doing more cases? And if we do we need to target specific types of cases or tumors for future single cell studies?
Yeah, I mean,
give
actually,
us the biggest bang for our buck, I guess.
Yeah. I think if you look now, most single cell sequencing studies are quite small. I mean, quite large in terms of the number of cells that you sequence per patient, but quite small in the number of patients that you actually analyze, within a cohort. That's mostly dictated by cost, also by the fact that it's hard to run a lot of experiments in parallel. I think now with some of the newer methods to do sample multiplexing, this is going to change quite a bit. So there are hashing methods. There are methods like multi-seq and other methods. We're actually developing some methods as well for some of the single cell DNA analyses so that you can pull samples together. I think now people are more routinely pulling together, 10 samples or so for one experiment. So I think it will be possible soon to look at, hundreds of patients at a fairly reasonable cost. So I think, yeah, that will be important to do. And then, a lot of calculations have to go into, I think, how many cells you can sample from an individual and still get a representation of the cell types of interest. You'll have to do some calculations there about what percentage of certain immune cells that you might be interested in might exist in the sample. But yeah, I think that's one big change, I think, in the field. It's, started as we could only sequence a few cells at a time or a few dozen cells. Now it's gone up to, you can sequence, thousands of cells, but you can only do that for one sample at a time. And now I think the methods finally exist is to, pull together 10 samples or more samples and kind of run these high throughput assays.
Mm-hmm. I realize we've been talking about the high cost of single cell sequencing, but I don't think we've said what that is. We have a question asking what would be the approximate cost for a single cell sequencing?
So a typical single cell RNA sequencing experiment, I mean, what we charge in the core, which is roughly equivalent to, I think, what most people pay, is about $3,000. And then there you would sequence 5,000 to 10,000 cells. But, with hashing methods, you could pull 10 samples together and, sequence 1,000 cells per sample if you would like to. And that would, bring the cost down quite significantly.
Thank you. We have a question about Copycat. So have you inferred trees or tumor evolution? And was it done directly by the use of Copycat, or was Copycat used for clustering and then on top of that trees were inferred by some other approach?
We did infer trees actually in that study. If you're interested in going to take a look at it, I didn't present the slides here, but you can use the data, so either the single cell copy number profiles, or you can do a consensus across the kind of clonal genotypes, and you can build neighbor joining trees or maximum parsimony trees and try to understand the relationship there. It's not the best data, I think, for really doing that. I mean, it's better to use genetic or genomic data like from single cell DNA sequencing. But you can get some ordering, I think, of the clones that might have appeared during evolution. And the great thing is then you can tie that to the phenotype, which is the RNA data, and really understand if there are 2 or 3 or 4 different genotypes, how are the expression programs different there. So I think that's the main advantage of using an approach like this. But yeah, absolutely, you can build trees using that data, and we did actually publish that in the study that came out recently.
Great, thank you. We have a lot of questions, but I'm afraid we are running out of time. So I think I apologize to everybody. We didn't get to all the questions you had, but thank you again for a very nice presentation. I'm going to turn it over back to Leah to give some wrap-up announcements. Thank you, Dr. Navin.
Thanks.
Right. So thank you, everyone, for attending today's SeqSpace webinar, and thank you for Dr. Navin for really a fantastic webinar. I think it was just a great overview for our group. So we welcome everyone's feedback to inform future webinars. I also want to announce that our next SeqSpace webinar is scheduled for May 4th from 3 to 4 p.m. Eastern Time, and I'll feature a presentation on predisposition genes and breast cancer risk in African American women by Dr. Julie Palmer, and we ask that you register now at our website. So thank you for really a great webinar, and we'll see you next time. Thank you.
All right. Bye, everyone. Take care.
Thank you.